Is this your first time? Okay. My dad was just
classes in
I wish I would have taken college courses. I school that they told me that my safety and I do is the way to go. So we did courses.
chemistry, biology, US history, world history.
Hey, they didn't take my chalkboard away. So sorry, JC. Sorry. Let me see if we can do this. Maybe. That'll help. There we go. Okay. Now it's not terribly in your way, or is it a lot in your way? Okay.
We have until tonight to do the polymer guessing, correct? Is that still open?
Sure. Yeah, I haven't done it. I haven't done anything to change that. I assume it's still open, ready to go. So yes. I like your yellow today. It's cheerful. Yep, it is. It is. I hope everybody had a fantastic weekend. I know it was nice just to have a little bit of downtime, and I hope you got some rest and maybe we're able to take a little bit of a break from micro studying. I unfortunately got a little bit behind myself, and so I haven't yet gotten through all of the hypothesis resubmissions. So I've been working on midterm grades and getting those submitted. And the good news is that this morning I finally got them all finished up, and nearly everyone is passing. Literally over 90% of everybody is passing. And so if you want to know what that exact midterm grade is, all you need to do right now is add your lecture grade and your lab grade, and know that those are up to date at this point. So that's kind of cool to just see that everyone is thriving. Almost everyone is thriving. So that's a really good thing to see. We've had one exam review session that went really well on Friday. So those of you that came on Friday, hopefully you feel a little bit better about the exam. And those of you that plan to come today from 3:30 to 4:30, we'll run another exam review. And then if you can't come today from 3:30 to 4:30, come tomorrow from noon to 1, and we'll do a third exam review session. So I'll hope to see you in one of those 3 sessions. And I think only one person last Friday said that they weren't able to attend any of those sessions. So please, if you are in that boat, let me know, okay? And we'll make sure that we meet separately. So otherwise, we're looking towards the 6 homework, getting that done. And by popular demand, we have, actually, I guess just by one single request, we've moved that due date to Thursday. I also went ahead and just planned to have the due date for the next homework on Thursday. So do make note of that as well. So that should keep things spread out in a nice way, right? Okay, so I think that's everything I had to say lecture-wise. However, lab is going to be heating up here pretty soon, because starting tomorrow, you will get your 2 unknown bacteria. So you will literally get handed 2 tubes, one labeled A and one labeled B. And the A is a presumed gram-positive, the B is a presumed gram-negative. However, you will be verifying, and then based upon what you see and the clue that we hand you, for example, it may be isolated from a patient with bloody diarrhea, and you'll be like, okay, I've got something to go on here, and you'll go do a little bit of research, and you'll come back with a hypothesis. So next week, next Tuesday, you'll have made 2 hypotheses grounded in background information and research, and testable. And then you'll also be proposing an unknown ID plan. That is to say, we want you to tell us how you're going to go about identifying this unknown. What are going to be the important biochemical tests for you? How are you going to use your newly acquired profiling skills to determine how you'll go about identifying these bacteria? So this gets really fun, and I hope that you'll dive back into the lecture material from the last week's lecture material for lab, that is, the lab intros, so that you know what to do when you get presented with that unknown ID plan assignment. Any questions on anything announcements-wise, or just maybe something that was cool this weekend? I keep forgetting, I need to post that beautiful image of the Starry Night, Van Gogh's artwork, famous artwork that they recreated on bacterial plates. So it's absolutely beautiful. I'll try to send that onto WyoCourses so everybody can see that. It's so cool. Okay. We will jump in because things were getting real last time as we talked about replication, and we had been looking at the replication fork and what was occurring at the fork where there were complexes called replisomes running bidirectionally around a circular chromosome and replicating the DNA, making new copies. We looked at the fact that DNA polymerase, the enzyme, is the enzyme, the race car enzyme, responsible for making new DNA by reading the old and incorporating building blocks to make new DNA. And we had said that this DNA polymerase enzyme is very much like a race car, right? It goes super fast, 1,000 nucleotides per second, and it also is incredibly accurate. It has its own proofreader function that allows it to be really, really accurate. So I want you to consider this first question. Despite being wicked fast and awesomely accurate, DNA polymerase is what we might term a needy enzyme. What three things does it require in order to synthesize new DNA? And I think Boris has our first answer. Oh. Yeah, exactly. Such a wonderful question. Remember that, so you did also answer this, right? Because he gave us number one, which is DNTPs. We know that it needs building blocks. And then at the same time, Boris's question is, do these provide energy? Does anyone want to speculate on that? Like when a DNTP is incorporated in the polymerization, does that provide energy? Yeah, Aaron. You got it. So it only needs one phosphate. I love how you said that. So in fact, it ends up being just a monophosphate nucleotide that gets incorporated. And so that cleaves one high-energy phosphoanhydride. It also leaves a PPI, which is two phosphate groups linked to one another. Those generally spontaneously hydrolyze as well. And that does provide a lot of energy. And yes, the answer is yes. And so we can say that this is an incredibly endergonic process, consuming DNTPs like crazy, not only as building blocks. What a smart, way to go about life. It not only provides the energy, but it also provides the building blocks. Has anybody thought about the second two things that it needs, Corinne? It needs primer. Nice. So we can actually say that in two ways. Primer is kind of the official way to say it. But what is DNA polymerase getting from the primer? Like, I should say, what is it building off of on the primer? Yeah, the 3' hydroxyl. Excellent. So in the primer, it's got its start site. 3' hydroxyl. So one of the analogies that I like to make is that there are certain race cars that won't start without kind of pushing them and getting them started by pushing them. This is what DNA polymerase is like. It's like a race car that cannot start itself. It has to have a 3' hydroxyl to begin the synthesis from. Otherwise, it can't go. Now, what's really interesting about this is that RNA polymerase is not the same. Another analogy that I like to make is that DNA polymerase is kind of like a downhill skier versus a cross-country skier. It requires the chairlift to get to the top of the hill, whereas a Nordic skier doesn't require chairlift. It's more like RNA polymerase. We just ski up the hill too, right? So this is the difference between DNA polymerase and RNA polymerase. DNA polymerase requires 3' hydroxyl. RNA polymerase can just cold put 2 nucleotides together. That's going to be important here in a minute. Yeah. You mean, are you talking about DNTPs as cofactors?
No, just like activators.
Yeah, you're confusing replication with transcription. And what I will say is that the site on the DNA is the sequence that's there in the OREC triggers, accumulation of all of the needed molecules within the replisome. It's site specific, yeah, yep. It's a recognition of, yeah, it's pretty cool. Okay, what's last that we're still missing here? A template, excellent, Mary, yeah. Great job. So there is no way for DNA polymerase to know how to add the next NTP unless it knows what to pair it with. So for example, if there's a T in the template strand, it knows that the next DNTP to add is DATP because T pairs with A, right? Okay. So that's what it tells it what to add. Let's zoom in and look closer at the replication fork. And we should talk about one of the favorite enzymes that actually precedes the replisome. So there's upwards of 40 components of a replisome, but there's also components that are outside of the replisome that will actually precede it and do some of the work for it. So as I said, DNA polymerase is a very needy enzyme. It also needs another enzyme called DNA helicase. Helicase literally runs out in front of it on the DNA and it unzips the genes. So I totally forgot to wear my t-shirt today, but I have this sweet t-shirt that says, I wish I was DNA helicase so I could unzip your genes. So it's a pretty sweet enzyme, right? It's out there in front. It's sort of forging away. It's unzipping the genes. The genes have to be unzipped in order to open them up for pairing of the template with the newly synthesized DNA. So that's what DNAB is, a particular helicase found in E. coli. Recognize too that topoisomerases, which are known in E. coli, one specific one is DNA gyrase. These are required to stop tangling, essentially, because at the replication fork, the unwinding can be wicked fast. And so tangling is a problem that can occur on that replication fork. Topoisomerases stop the possibility of that tangling by relieving the supercoiling ahead of the replication fork. So DNA polymerase literally has to have the red carpet put out for it. It needs to have things ahead of it so that it can race car drive through without any kind of problems. DNA helicase and topoisomerase are like those that preceded and set out its red carpet for it. Now recognize that this is the OREC right here, and we're zooming in on that site. So if we were to add to our drawing the replisomes, you would see one large replisome here with its many components, and it would be heading this way. And then you would see the other one backed up next to it, and it would be headed this way, bidirectional. Now, as it does so, the interesting thing is that this replisome synthesizes both this strand and this strand of DNA. So if we were to draw that out in an anti-parallel way, we know that this would be the 5' end, and it would be synthesizing a new strand complementing the black template strand in this direction. That is, this would be the 5' end, and this would be the 3' end. Likewise, on the opposite side, we know that it would be synthesizing this other strand, sorry, bad strand, in which this would be the 5' end, right, there, and this would be the 3' end, okay? Now, likewise, this replosome over on the right would be synthesizing 2 strands as well. This, the 3' end, this, the 5' end, and likewise 5', 3'. What? Kirsten's like on this. She's like, Rachel, there's a problem. What's the problem?
Well, I was just wondering why it's like opposite on the side. Like, why wouldn't it be 5, 3, 5?
Well, because it's always anti-parallel, right? Because this is 5 to 3, so to speak. And so you would see that the strand that would be synthesized is always anti-parallel, right? Yeah, yeah. But there is a problem here. Anyone picking out the problem? Yeah, good, Adam. So recognize that this, where we see DNA being synthesized, 3' to 5', this is not a thing. This does not occur, right? So this does not happen. Why not? Yeah, because DNA polymerase can only go in one direction. Why can it only go in one direction? What does it always add new nucleotides onto? Or AKA, what does it need on the primer in order to make new DNA? What has to be present? 3' hydroxyl. You got it. So this is not a thing. The only direction that DNA polymerase can synthesize is 5' to 3', where it adds new DNTPs onto the open 3' hydroxyl. So this works great. There's an open 3' hydroxyl right there. DNA polymerase just zooms along, adding new nucleotides to that open 3' hydroxyl. Same thing here. This is totally possible. New nucleotides onto the 3' hydroxyl. But we know that this strand and this strand can't possibly be synthesized that way. So we have to think about a creative way that DNA polymerase and this replosome can somehow make these strands that appear to go in the 3' to 5' direction, even though they do not. So let's talk about that, and it might help to have the not Rachel's messy writing version here. So DNA cannot be synthesized in that 3' to 5' direction. They can only go 5' to 3', adding on to the open 3' hydroxyl all the while. So what this means is that DNA polymerase has to get super creative, or the replosome as a whole, in order to somehow make this strand here. And does anybody remember what we call this strand here that has to be synthesized in a discontinuous manner? Lagging strand, good. So this lagging strand literally has to get made in discontinuous spurts. So this strand, the leading strand, it gets synthesized continuously, no problem. But the lagging strand has to be synthesized discontinuously. So there is a way that I like to think about this, and if this analogy works for some of you, it might be helpful. Does anybody knit at all? Yeah, I love knitting when I have time to do so. But if you knit with 2 colors in your hand, you have one strand that's maybe your base color, right? And you just keep continuously incorporating it like this. But in your other hand, you have your counter color. And whenever that counter color comes up, you literally have to take it backwards like this and wrap it around the needle and incorporate it that way. So it's kind of this weird thing of like continuous main color and then wrap back around discontinuous with your other color. This ends up being what DNA polymerase and the replosome does, is that it literally continuously synthesizes the 5' to 3' strand, knitting like the main color. But then with the lagging strand, it literally wraps it backwards around through the replosome so that it can synthesize in short bursts in the direction that it knows how, the 5' to 3'. So I'll show some, I'm going to show lots of visuals today so that you can visualize this, but that's one that helps me think about it. Yeah. Right. So, I mean, that is a super awesome evolutionary question. Like, why instead don't we see a situation where two replisomes each do the leading strand and they go all the way around, right? Seems like that would make more sense. It's just not how that evolved. Yeah. But I love the question. So I'm going to draw this on the board so that everybody gets a sense of what's going on. We know that everything is cool for the leading strand. So what I've done on the board is I've taken the leftward replication fork. We're leaving out the right one for a minute so that we can just focus in on the left-hand one. And on this, we know that this is our leading strand, 5' to 3'. And all of that is good, no problem, easy peasy, right? But the lagging strand, things are not so simple. So what ends up happening, and of course I can't draw this on the board, but this strand ends up wrapping backwards around through the replosome, and we'll see that in a minute. But even before that happens, one of the components of the replosome, so here's our large replosome here with various components in it, okay? And it's kind of encompassing this whole area. And so as it heads this way, and it's easily making the leading strand, the other thing that it does on the lagging strand, so if this hand is the leading strand and it's synthesizing continuously, what happens with the lagging strand is that there's an RNA polymerase called primase inside of that replosome machinery. And as this goes like this, leading strand being made, the primase makes RNA in these short little 10 nucleotide primers, and it does it like this. Okay, so watch. Right? Does that make sense? Like it's actually going backwards with its little primers, but it can do that because they're so short, right? They're teensy tiny little primers. So let's add in our teensy tiny little primers. They're only 10 nucleotides long. But we know they are in the CMID right direction, where it looks like they're getting made 3', 5', right? Even though they weren't made that direction, they were made 5' to 3', but it's just that they're so tiny that it could go backwards like that, the primase. And remember, this is an RNA polymerase, so it doesn't need to have a primer itself. It makes the primers. So these little guys are not DNA, they're RNA, okay? So they have the hydroxyl on the 2' carbon, they're RNA. Now what happens is that this strand gets looped around back into the replosome, right? Weird stuff. And that exposes the open 3' hydroxyl for 3' to 5' synthesis of DNA. So the DNA is like, woohoo, there's my primer, my open 3' hydroxyl. And as it's looped backwards in there, it synthesizes like that, right in that direction. And this one would have been first. And it fills in DNA in there. And does anybody know what these little fragments are that start with an RNA primer and end with DNA? Okazaki fragments, right. So those are little Okazaki fragments. So let's jot this down in our notes, making sure we recognize the action that has to occur. Primase is the name of the RNA polymerase that makes the primers. It makes those little primers along the template strand. And then DNA polymerase synthesizes the complementary DNA, just the same old polymerase that's in the replosome. And we get our Okazakis. What happens next is fairly interesting. There's another DNA polymerase that is actually capable, it has a 5' to 3' exonuclease in it, so it's actually capable of removing these little primers. And it does. It just takes out these little pink primers. Okay, be gone, little pink primers. Okay, and now it fills in DNA in those sections. So look, for all intents and purposes, it looks like this strand was made 3' to 5', even though it never was. It never was. It was always made 5' to 3'. So writing that down, as DNA polymerase adds nucleotides to an elongating Okazaki, eventually it reaches the point of the previous fragment. That is, the Okazaki fragment hits the butt end of the previous primer. And then DNA polymerase 1, another DNA polymerase, comes in and cuts out those RNA primers, and it replaces them with DNA. Of course, what enzyme was needed to just form those 3', 5' phosphodiester bonds in between the little fragments, to just seal it up? Yeah, knit it together, ligase. Ligase will form the 3', 5' phosphodiester bonds between the fragments. Sweet. So admittedly, this was tough for me to understand until I built my own model. So I'm kind of a slow learner. It takes me doing a lot of stuff before I visualize this. So let me show you the model that I ended up building. And I'll try to turn it, let's see if I can turn it so that it looks like to you guys the one that we've drawn up. Well, it's going to be kind of tricky because I want you to see. I'll do it like this. Okay, so this, the styrofoam toys right here, this is representing the Reckless Ohm. And as you can see, this is one of the replication forks. So we're just looking at one. This, what's going on here? That's, yeah, so the purple is the new DNA, and this is the leading strand. So as the Reckless Ohm heads down the replication fork, it just adds new nucleotides to an open 3' hydroxyl right here, and this is the leading strand, no problem, right? But this is the lagging strand. And on the lagging strand, what we can see is that the fluorescent green represents the little RNA primers. So every 1,000 nucleotides, there's a short 10 nucleotide primer laid down. So now notice that this is the 5' end and this is the 3' end. So DNA polymerase can't add onto the 5' phosphate. That's not a thing. So this gets flipped backwards like this so that the 3' hydroxyl, okay, so check it out. Here's the 3' hydroxyl there, right? Flip backwards like this. A lot of finagling. And then from that open 3' hydroxyl, new DNA, right? Purple, visualize purple getting added and added and added as the Reckless Ohm heads that way until this hits the butt end of the next primer, right? And then it drops it. And then it'll do that again when it hits the next primer, okay? Does that kind of help you visualize it, Erin? Yes. DNA polymerase 3 is the primary DNA polymerase that builds the leading strand, and it builds Okazakis. DNA polymerase 1 has a 5' to 3' exonuclease. It removes the primers and fills them in with DNA. Great question. And now if we start looking at eukaryotic systems, we start looking at even greater complexity to that. Beautiful. This is just an older picture from your book that tries to show that in a different visual way. You can see the RNA primer here, the way that it's flipped it back around to make the lagging strand. And then likewise, you can see the continuous, easy leading strand synthesis. This is a picture, or this is a cartoon from the Walter and Eliza Hall Institute of Medical Research. And here it is, DNA helicase unzipping your genes. Okay. And then this next one, be ready for it. This is actually a really cool computer animation. I want to make sure everybody can see this, showing the Reckless Ohm heading down the fork. And you're going to be able to see leading and lagging strand synthesis. I'm going to do my best to make sure that I narrate the whole thing, okay? So if you want to go to this site, it's just Towers Hughes Medical Institute, Walter and Eliza Hall Institute for Medical Research. Here we go. Okay, it's headed down the Reckless Ohm. This is DNA helicase unzipping the genes. This is the leading strand getting synthesized continuously. This is the lagging strand getting synthesized discontinuously. You can see it picking up from the primer, doing the Okazaki fragment, hitting the butt end of the next primer, dropping it, and picking up again, and continuing that discontinuous synthesis. Okay, so pretty cool, right? And this, I know, this animation can be found online. Definitely go on and check these. There's some other ones that are really cool as well. So hopefully, all in all, with all those methods, everybody is feeling a little bit better about visualizing replication. And on that note, we are now moving away from replication to transcription. So does anybody have any broad questions about replication? Okay, so transcription, broad definition means making what from DNA? RNA, right. Making RNA from the DNA code. That's pretty easy, but the hardest thing about transcription, and this is something that Boris's earlier question gave light to, it's all of the lingo that goes around transcription. For example, the name that we give to the landing pad where it all begins is called the promoter, and that's shown in my picture in blue. And then the gene is generally dictated as what we call the +1 transcription site to the termination site. So the functional unit that encodes for the product actually is given numbers, and that's kind of confusing. We also then name both strands of DNA. So the top strand is always referred to as the coding strand or the plus strand. So here, the top 5' to 3' strand of DNA called the coding strand. Lingo, lots of it. That means that the bottom strand, or the 3' to 5' strand, one name that we know that it must necessarily have is the, not positive, but negative, right? And this actually is really important to remember because we talk about plus sense DNA or plus sense RNA all the time, and it means that it goes in the 5' to 3' direction. So the 5' to 3' strand is always termed plus. The 3' to 5' is always termed minus. The other name that's given to it is that it's called the template strand. And that actually makes a lot of sense because that means that it's the strand that RNA polymerase reads in order to make the RNA. So the template strand is the one that gets read, okay? So the bottom strand, we know it's the minus strand. We also now know it's the template strand. But more lingo, because the gene is said to begin at the +1 transcription start site. That is quite literally, we have just decided that the nucleotide that starts the gene is going to be called +1, which means that then the next nucleotide is +2, and the third is +3, and so on down the line. Interestingly, then, when we start talking about that blue region, that is where it all gets started, everything to the left of the +1, that is in the blue, everything to the left is -1, -2, -3. There is no 0 nucleotide. So that is lingo to help us orient ourselves on the gene construct. Transcription terminates at a site that is aptly called the transcription termination site. And we'll talk more about what kinds of sequences are found there. So let's make sure we can get probably what is the #1 take-home message of this entire gene expression unit. And I'll probably say it like 500 times. You guys are going to get really, really tired of me by the time we hit the end of transcription. But if we know that the RNA polymerase enzyme lands right here, this is like the RNA polymerase landing pad, and the minute that it hits the +1 sequence on the gene, it starts reading and it starts making RNA from the DNA. Which strand is it reading once again? The template, good. So it reads the purple strand. So as RNA polymerase is headed down this gene, it's reading the purple strand, which means that as it builds RNA, what does that RNA end up looking like? Yes, let's say it again. As it builds RNA, what does that RNA end up looking like? The coding strand. Okay, this is #1 take-home message today. Write it on your hand or something as you leave so that it's there for the rest of the day and you're like, I will always know this. The RNA looks like the coding strand. Only what are the little differences? U's replace T's, and it's single-stranded. Excellent. And lastly, what's going on at the 2' carbon? There's a hydroxyl, right? So that's really the only difference. So what's cool about this is you can dive into any literature, and I suggest doing it right now if you want to, and look up gene sequences. And if you're given, and most of the time you will be, given the coding strand, you can immediately know the RNA sequence. Because all you do is run through and replace every T with a U and know that it's RNA instead of DNA, okay? So this is a really, 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 really important take-home message. Is that the RNA has the same sequence as the DNA, except that U's replace T's. And you could also make note to yourself the beautiful things that Brian came up with. Yeah, it's single-stranded instead of being double-stranded. And also that there is no 2' hydroxyl on the DNA, but on the RNA, of course, there is. So those are two of the other small differences. But this is such an important thing. Okay, more lingo. You knew there was going to be more because this blue landing pad where transcription all begins is called a promoter. At the promoter, this is where a lot of things associate. And I think this is partially where Boris's earlier question rooted, because there are a lot of what we call transcription factors that all associate in the same promoter region. And all of these protein complexes that are needed to make RNA happen all come together at this promoter because of specific sequences. So it's not box, right? It actually has unique nucleotide sequences that are recognized by the transcription machinery. So at this promoter that serves as an initiation site, we see all of the transcription complexes assemble. Now, this particular picture is a pretty simplistic one, but what it does show is it shows that one gene is under control of this promoter. In bacteria, we often see situations where more than one gene is controlled by a single promoter. So picture this, a situation where we have poor instructor handwriting, where we have a 5' to 3' strand, just as we're seeing in the other picture, and a 3' to 5'. And we recognize that we may have our promoter here. I'm just going to label it P for simplicity. But downstream from our promoter, we may very well see more than one gene. So we might see genes 1, 2, and 3, for example. This sort of situation where there is a promoter controlling 3 separate genes is given a special name. We actually call this an operon. So an operon is a construct in which a single promoter controls the expression of more than one gene. Now, does anyone know of a very famous operon? The lac operon. And in fact, the lac operon does have 3 different genes on it. So those of you who've maybe taken genetics have seen that before. So this is called an operon. You could get many, many more genes too. You could get 5 genes. And what happens when an operon is read, so we know that RNA polymerase could bind to the promoter with all of its ability to read down this construct, and it would head down, and RNA polymerase would read the 3' to 5' strand such that the RNA would look like the 5' to 3' strand and would carry the information for gene 1, gene 2, and gene 3 on this single-stranded molecule that had a sequence that was the same as the coding strand, only U's replaced T's. And the special name that we give for this RNA when it carries the information for more than one gene, we call it, well, how about you tell me, if there's more than one gene, what would be the term for more than one or many? Polycistronic. So this is a polycistronic RNA. So it carries the information for more than one gene. So in eukaryotes, we usually see monocistronic genes where they only have information for one gene, but in bacteria, we commonly see these polycistronic mRNAs. So then you're wondering, well, what is it about the promoter that makes RNA polymerase and all the transcription factors associate there? It's the sequence, and the sequence has a consensus. The sequence is 2 regions that are found by comparing multiple organisms in their upstream region. Okay, that was just a lingo term. What I mean is in the region where the promoter is to the left of the +1 start site. We see these 2 areas. One of them is the -35 region, and that's this region here. And its consensus sequence is TTGACA, or said TT-GACA. The other consensus sequence is at -10, and it's called the Tata box, or the Pribnow box. So these 2 regions are the consensus sequences that indicate to RNA polymerase that this is where you want to be. In fact, RNA polymerase has a special subunit, a promiscuous subunit, in fact, called sigma. And sigma, when it sees TT-GACA and Tata-AT, it's like, well, hello, good looking. And it stops right there, right? It holds on to TT-GACA and Tata-AT. It really has a high affinity for these 2 sequences. So the closer the promoter sequence is to ideal, TT-GACA and Tata-AT are ideal, the more affinity RNA polymerase sigma subunit has for those sequences. And in fact, the more frequently the downstream gene or genes would be transcribed. So if these are really close to ideal, that is, we call it a very strong promoter, then the genes downstream are very frequently transcribed. But let's say, for kicks and giggles, that this was AAC-ACA. Well, that starts getting pretty far away from TT-GACA. So the RNA polymerase enzyme would be like, I think you're good looking, but I'm not so sure, right? It doesn't have as high affinity for that kind of sequence. We would call that a weaker promoter, and it would be transcribed much less frequently. We're going to go ahead and practice that so that you can see how good you would be at being RNA polymerase and how well you would recognize a strong versus a weak promoter. And these ideal consensus sequences have just been determined by comparing across lots of different promoter sequences and finding the most frequent nucleotides in those positions. So let's do a little bit of practice on this. Genes downstream of which promoter would be transcribed the most frequently. So take a moment and think about which one of those is the strongest promoter. If you're RNA polymerase, which one's the most good looking to you? Who's feeling bold and rested this Monday? Think you found the strong one? The third one? Somebody, okay, somebody's saying the third one. Let's go through and look here on the third one. TT Gaga. Okay, so there's only this difference in the -35 sequence. And Tata at, okay, there's a difference there, right here. So there's only two differences. How about the number of differences in the second one? There's a lot, okay. And then how about the first one? Even more, okay, good. So it looks like the third one would be the strongest promoter. Nice job. I have a question for you to practice it all. So we'll pull out and pull up. So all the following accurately describe either prokaryotic replication or transcription, except during replication, Okazaki fragments are formed during the synthesis of the lagging strand of DNA, but not during the synthesis of the leading strand. The next deoxynucleoside triphosphate that DNA polymerase would add to the currently synthesizing strand would be DCTP. A promoter with the -35 sequence of TGCAGA and a -10 of TATTGT is stronger than a promoter with a -35 sequence of TT-GAGA and a -10 of TATATT. The coding strand of DNA has the same sequence as the RNA, only U's replace T's in the RNA. Okay, so pull on in here or get your answer because my poll doesn't have all the words on it. So, oh, it's mostly the resolution of the projector that's an issue. I can just tell you what these are. Like this is cytosine, this is guanine, this is thymine. So cytosine, guanine, thymine. It looks like we're having way less of a war with this one today. That's good. Answers are accumulating as C. So this is correct because notice that TT-GAGA and TATA-ATT are actually very strong. So this latter mentioned promoter is much stronger than the former mentioned one. So this is our exception. But do understand that DNA polymerase would add to the open 3' hydroxyl, as it always does. And so it would pair a DCTP with the guanine, with the G here. replication, Okazaki fragments are on the lagging strand. That's correct. And then finally, I hope everybody is pretty cool with this idea that the RNA looks like the coding strand, only use replace T's. Promise I would say it a million times before the end of the day. So I used this term a couple of times, but I did not do a good job of writing it down. So the leftward side of a gene, the side to the minus side, where the promoter lies. And in fact, this is entirely dictated by the location of the promoter. The promoter side of a gene or genes is always upstream. The termination site of a gene or genes is always downstream. So it's actually entirely dictated by the location of the promoter. Upstream will always be where the promoter is. This is important because as we kick off, and actually I think we have time to look at this really quick. As we look at a plasmid, okay, so this is a made-up plasmid. We'll call it the P-fun plasmid. We've got two genes on this plasmid, gene 1 and gene 2. And the promoter for gene 1 is labeled promoter here, and the promoter for gene 2. Let's understand the terminologies of upstream and downstream. So on gene 2 here, the upstream is this region. Whereas for gene 1, upstream is this region. For gene 2, this is downstream, right? And so this is kind of all dictated by the gene. Now let's go through and see what the RNA would look like. So if RNA polymerase came through, if RNA polymerase came through and read gene 2, which strand, the red or the blue, would it be reading? So it's reading the red. So what would the product look like? It would look like the blue. So the RNA would end up looking like the blue strand, and that's a horrible RNA, but it would look like the blue because it's reading the red. Now what about if RNA polymerase comes up and reads gene 1? We know it's going to bind to the promoter for gene 1, and it's going to read this way, right? It's going to read this way down gene 1. What strand will it read? The blue, so it'll end up looking like the red, okay? So it all has to do with where the promoter is located, what the ending RNA looks like, okay? And what is the coding versus what is the template is all relative to where the promoter is. All right, have a wonderful day, and please remember that RNA looks like the coding strand, only use replace T. Okay, get out of here.